Hello, I'm going to take you through the exercises on protein sequences, and here's the part on Uniprot. 1. What is the Uniprot ID for human hexokinase? So to do a search, we open a new tab, go to the Uniprot webpage and enter the text. So we're looking for human hexokinase 1. When we look at the results, we can see that human hexokinase 1 and its isoforms show up at the very top. So we can go to the entry, which happens to be the Uniprot ID. So we can copy it from here. It is P19367. And that is what we can enter into our template. 2. How many amino acids does the protein have? Going back to our template, where would we find the number of amino acids? So typically the best place to go and search for the length of a sequence is to go to sequence. So we can see here's a tab called sequences, and if we scroll down or go there via the shortcut, we can find the length indicated on the right hand side, and the length is given as 917 amino acids. That would be the piece of information that goes into our template. 917 amino acids. 3. How many glucose 6-phosphate binding sites does this protein have, and in what positions are they? Positions referring to the number of the amino acid in the chain. So let's have a look. How would we go find about binding sites? Typically such information is summarized under function. So if we scroll to function and then have a look at what is there beyond the reaction that is catalyzed by this enzyme, we find the table sites. And under sites, we can also do a textual search. Glucose 6-phosphate. Going back to sites, we can see that the highlight indicates that we have 1, 2, 3, 4, 5, 6 binding sites for glucose 6-phosphate in this protein. And we can also look at the binding sites and pick up the numbers as 209, 232, 449, 657, 680, and 708. Go back to our document and feed that in. So we have 6 binding sites, and respective numbers are 209, 332, 449, 657, 680, and the last one was 897. You can specify that these are at position and so on. So let's see the last question. Last question is 4. Which amino acid residues form the binding site for ATP? Now that's actually simple because binding sites for ATP are in the same table. So if we look at ATP, we can see there's one at position 30, the second at position 3, 4, 5, and the last one at position 680. So let's bring this into our template, and the binding sites are amino acid 30, 3, 4, 5, and the final one was 680, which coincides with one binding sites for glucose 6-phosphate.